That's a big crab.
Come down, Jaden.
Copy.
Yeah, these were really fun to watch in the beta camera video, because you could speed up the video and just watch them running around, chasing everything else away from the bait, fighting with each other.
Push in there a bit for me.
What kind of carp is this?
This is a Chaseon microniscus. And it looks like the carapace is about 15 centimeters long. Those are
Right.
some serious pinchers.
Yeah, science is happy, thank you.
All right.
Is that a little hatchet fish?
Yep, I think that's a hatchetfish.
You can go ahead. See if we can get over the hill here. We're just putting that lens on top of us, so I got some leash to drop down the other side there.
All right.
So I gotta admit, I would have expected more. I mean, not that there's not a good number of corals up here, but I was thinking there would be more. And I'm interested at how the primnoids are dominating up here. Like there's a Walteria sponge, we're seeing more of the kind of encrusting and smaller sponges, but in terms of corals, it's vast majority are members of the family Primnoidae, with a couple Anelopsamia thrown in here. But can we look at this sponge, please?
Man, I think I just saw like 5 or 6 Oreos in one frame. That's quite a lot.
Come down,
My
good.
goodness, they're everywhere. 1, 2, 3, 4.
Do they travel in groups? Is this
Yeah, you can push in
common?
there. Hold that.
I don't know. I mean, in general, in the deep sea,
Okay.
we're not used to
Push it a
seeing
bit more.
large groups of fish. And I guess okay. This, it kind of looks like this,
Oh, you want to come
Sponge.
down here, pull me off.
sort of. Yeah.
Atalanta's altitude is single.
Yeah, I see.
We would
Yeah.
like to collect that sponge.
Roger.
Bridge nav, 15 meters, 100.
Can I go in?
got a light if you want it.
You can push in again. Roger. You're putting that latte right over the Hercules, are you? Thank you. I guess we have to grab it since our slurp is wounded.
Yep.
Right. Might wait for a minute till we get Atlanta a little closer here.
Sure, that's fine.
If I get distracted, we're single digits right now. Unless you keep an eye on it, Jaden. But if you do come up, just come up really slow.
Copy.
This will probably tear, which is expected.
You just want a piece of it or the whole thing?
Take the whole, go for the whole thing, but I think it'll tear off its base instead of pop off the ground, is all I mean.
Let's play with the grip force here so I get it super soft.
Kenlin, this is the tentative ID.
Before I try it, you can come up super slow to double digits there.
Copy.
You'll get a good shot of her because you're coming in. So. That's probably good for now, Jade.
Copy.
Thank you. I'm going to spin around just a little bit first to see if I can get the vehicle to stop rocking around. Go wide for me. Put our head into the breeze just a little bit more. While I'm up, Jaden, you can come up just a little bit.
Copy.
Just a little. Look down some more. That's better. Stable platform. Okay, you can zoom in again for me. Right, get a tight shot of it there if you want. For
Very good when you're good.
right. Trying to get the manipulator parallel with the rock, and I'm having freeze fail, multiple freeze fail. Touching the rock there, because I see the camera moving. And zoom out for me. Where are you going, Herc? What's happening?
Definitely picking up some midwater fish flying around here too, not just benthic fish.
Yeah, I saw a couple hatchet fish.
Yeah, there's one dancing around here. The odds of you being able to keep this intact, like fully intact, are pretty low. You're just probably going to have to grab and rip.
Ripping. Come on. So sponge, all right.
Yep. Can we get a really tight zoom on it and its claws and the claw?
Yeah, you can go full zoom there if you're not already.
All right. Where you want it, Quinlan?
D.
Can you get in a little box, or do you need a bigger
Yeah.
box?
No, I can get it in a little
Okay.
box.
All the way aft, then.
All right.
Can we look?
What do you want to look at?
The pink thing, if we're waiting here, or is
Right.
it already full?
Right, yeah, I think she's all the way tight there.
Okay, well, and that's all we got. Never mind.
You can zoom all the way in on the... Okay, I'm gonna open up, you see a benthic ctenophore float out, close it. Hey look, it's a bonus sample.
How did that happen? Well, that went into E, so let's call it another sponge in E.
You want to put this sponge in E
Yeah,
with it?
put that sponge in E with it. That'll be
Sponge and bonus sponge in
sponge
E.
and bonus sponge in E.
Okay, you can close a little bit. So
Was
the
that a
ABCDs.
piece of the original sponge?
No, that was the other. That was one
Okay.
of the other spikier ones we've been looking at.
It just
I just
jumped
literally
in.
jumped in the box. Like, I mean,
Well,
that was amazing.
That's atypical.
Yeah.
This thing gonna
So
sink or?
yeah, I should think. So I know no one on shore saw that, but as we
Well,
opened
they
the
probably
box,
did. Okay, close.
Oh yeah, if you're watching stream 3, one of the other sponges that was right next to us of a different type just broke loose and floated directly into the bio box.
I think only Josh has managed that before, the bouncing sample into the box. Okay, how are we in turns and stuff? We're good. You can go wide for me, thanks. See, we were gonna hop over the hill, weren't we?
Yep. I'd like to run over the hill to the south a little bit and see if there is a marked change, and then we'll proceed towards waypoint 5 in whatever we find is the heaviest coral area.
All right. Okay, Jaden, you can come down a couple meters for me, down to that single digits there. You're right on top of the hill, so let me run down the hill with my nose into the breeze here.
Copy.
See if Herc will hold it sideways.
Yeah. Black coral, still primarily primnoids. Another one of the urchins, another couple urchins. This should be loaded.
Yeah.
All right, well that answers my question, I think, as it doesn't change dramatically over on this side. So let's get as far down as you can go without moving the ship, and then probably head back towards the ridge.
All right.
Got it? Yep.
I got a little left to drop down somewhere.
Well, now it's picking back up again, but not too much. It's the same community as we're seeing on the ridge top.
Yeah, it's come down a couple more meters for me.
Alright. How low is safe to take the altitude?
If you touch, you're too low.
Yep.
Alright, can we take a look at the big coral though?
Roger. You can come down a couple more meters, Jaden. The weather's pretty calm, so I just need just a little more to. Yeah, that's probably about it there.
Okay, half zoom will be appreciated.
Yeah, you can go ahead and push in.
Great, that's good, thank you.
Okay.
Alright, back to the top, thank you. Look at all the midwater fish. Little nyctophids, hatchetfish.
Yeah, it's hard to say from the sort of here and there occurrences of them, but these midwater fish do get compressed against the top of these seamounts sometimes, either through lateral advection or their vertical migration pattern being interrupted by the topography of the seafloor.
Or both and, more
Both
likely.
and.
Big black coral here as well.
And a fish.
Can do pushing on the fish there.
Oh, so many fish to choose from. We've got 2 Oreos, a cusk eel, and a rattail. And another rattail. Actually, yeah, let's zoom on this small rattail.
Go ahead. Zoom in there while it's centered up. Okay. Okay. Go ahead.
All right, thank you.
I can turn the multi-beam so it's actually spanning the ridge there on the way up if you want. Okay, coming at you, Jaden. Can slowly come up as I come up.
Copy.
You said we were going to go west next? Roger. I'll slide around to your west there. Other west. Left west. Doesn't want to go that way. Wants to go downwind. look to your right a bit for me.
Copy.
So I know the pickings don't look very, it don't look like we have a lot of options, but let's all keep our eye out for a rock that might want to come with us.
Right.
Are we on the summit now?
We are basically on the
Okay.
summit, yep.
Okay.
We look at jellyfish, if you got the time.
Oh, yeah.
Yeah, so we just
Push
came
in
over
about
the top
halfway there
and we're
for me.
now just on the south side.
Okay.
And we're gonna move along.
There's that
Not
holothurian.
jellyfish, yep,
Push in a bit more.
Plagiaria. These are cool. So these are fully benthic holothurians.
Fully pelagic.
Pelagic, thank you. Words are hard.
So that umbrella-like structure is called a vellum,
Wow.
and that's what it uses to swim, analogous
Whoa,
to a
look at it, so
jellyfish's
cool.
bill.
Gonna drop the lasers.
Yep.
Alright, science is happy.
Alright, I'll let him fly into us here and get Okay, you can go wide. Where did we go? We went downstream 10 meters.
That Oreo down low really, for a second there, I thought it was an octopus. Like something about the ripply movement of the dorsal fin made it look like an arm.
Oh,
That's
that's
a
a
squid.
squid.
Where'd you see a squid?
Just went out of frame left. It's down by like the Orion. I don't see it coming back though. It was little.
I'll just drift down with the breeze a bit. I see him like up above us now.
Yeah, I think it's gone.
He's faster in the current than I am.
So we're obviously into a bunch of the midwater stuff. It's kind of trapped here on the thing. So where are all the anemones that we had yesterday?
Great question. I have no idea.
Yeah, I mean,
Might be a depth range thing, or was it about the same?
It's pretty similar. Yeah, I guess we were almost 200 meters shallower, but clearly we're seeing more midwater critters. We zoom cup coral, please, or anemone, or whatever that is.
Sure. Go ahead. Push in a bit there.
It's like an anemone, maybe a ceranthid. All right, thank you.
Okay, move it up.
here, please.
Roger. Go ahead, video. Push in. Good, thanks. I'm light years above it. Let me come down.
I'm not sure what this is. Asako or Steve? Do you have a sense of what this is?
Okay, can push in a bit more for him. Should be able to full zoom there.
I'd like to sample that, please.
Roger. I got any boxes left? Oh yeah, we got D.
We got D. Focus a little bit on more on the polyps, please. Perfect. All right, let's put it in the box.
Roger.
We have to leave that at just unknown octocoral. I'm really not sure what that is.
Yeah, okay.
Can go wide there on the. Find something else to look at. I don't know, maybe a little yellow guy or something. Just anything Brian wants to see.
Just zoom. We'll find something interesting, I'm sure. Just zoom in.
The whole world in there. Whole other universe.
Yeah. Look, we got 2 sclerotinians and another, whatever that thing is we just collected. And a brachipod. I'd say that's some kind of like the polyps almost look like a true soft coral, but it's got a skeleton, so it's not that. I'm not sure what to make of that thing. It might be some kind of baby bamboo, but I didn't see any hint of a node.
Okay, you can
You zoom out
push the
just a
box
bit,
out
there's a
for
fish.
me.
Hold on, they got their hands full.
See, this is going in the last little one, right?
Yep,
Yes.
Perfect.
flyby fish. I don't know what to expect for behavior on this one, Dan.
Roger. Should I go for the monkey and the monkey fist in the jar? Or
Try this. Let's see what happens.
all right.
It's sinky enough.
Better luck than sticking my big snaggle teeth in there. Okay, you can close it. Thank you.
Oh, that is sample number 73.
Now that one's going to be a mystery for a while.
That a little cup coral there we're looking at too?
Yeah, so there's two different types of sclerotinians. You got the cup coral here, and then this yellow colonial sclerotinian.
That's the brittle one?
They're both very brittle.
Oh, okay.
Yep.
But you're saying the one that we just collected is soft?
Yeah.
Okay.
Well, we're not sure how soft. So
Yeah.
there's the true hard corals, and then there's the octocorals that have proteinaceous skeletons that can range from very brittle to very soft.
You can
And then
go
there's
like
true soft corals that have like no skeleton. And I'm not 100% sure where that one we just, well, normally the true softs have a much fatter, like almost like an anemone type base on them, not like a branch and then have a branching pattern. At least ones one I'm not used to looking at, or like the one we just collected, but I can't even give you a family on what we just collected.
There's another sea spider.
You want your
So
Atalanta
how does it
screen
make you
back
feel
there?
when you like come across something like that? Do you like get excited, or
I'm okay.
are you just
I meant
like,
to
I
put
mean,
it,
I'm a bad taxonomist, so it's
Yeah.
pretty
I meant
normal
to
for
put
me.
it down
Okay.
in the other
Um,
box, but I missed.
But it can, certainly, when you get
Yeah.
something new. But I see a lot of things that are new to my eyes that other people are like, oh, it's that. Couple more big crabs.
Yeah, I think we're back on the summit here, or the ridge running.
Oh,
Run some ridge.
Be interesting to see the Norvit, but do they sweep down both sides, or is it too steep? Oh yeah. Sweet.
yep, definitely sees down both sides.
That's awesome.
The catch is going to be at altitude high enough to see down both sides effectively. Can't see what's living on it.
So I think I might, I don't know how you want to run the ridge, one side or the other, or pop them back and forth, or whatever side has more.
Yeah, basically whatever
Diversity.
side looks like. I would assume, see, current is basically running along the ridge from the west.
I think the Oreo is a good indicator.
Which side do you want to do, Harold?
Yeah, I mean, based on that look we did earlier, they don't look too terribly different.
I agree.
If it's like this, I can
So,
jump back
yeah.
and forth.
Okay,
We
well,
can
let's do
hop
that
back
then.
and forth every now and then.
You have a good sense for the... actually, if Atalanta brings his head to the left a little.
Picking up a lot more of the sclerotinians all of a sudden.
It's pretty much west, I think, Chris. Matches your
Yeah.
macro bathymetry.
All right, so just start heading to the waypoint, and you're going to follow
Sure.
microbathymetry. Is that the idea?
Sure, yeah.
Oh, all right. Bridge nav. Let's do 260.
That squat lobster just bailed from that coral. Look at this zigzag in the seafloor.
Yeah.
Come down a few meters for me, Jaden.
Copy.
This reminds me a little bit of a feature we saw south of Helena Baker, where it was just a field of Calagorgia, which is another type of primnoid. They were bigger, they were substantially bigger at the time, but the same kind of view of all the fans lined up in the same way. And I think it was like the whole field was like 150 meters by 200 meters of just like, a meter, meter and a half coral every half meter. It was amazing.
Wow.
That was a couple hundred meters shallower. That was like 300 meters, I think. Can we look at this one, please?
Sure. You can come down a few more meters while we're... the golden one there? Or?
Correct.
Okay, should be able to zoom there, maybe. Herco, hold still. You should be able to zoom right in. Hopefully I'm sitting on the seabed there. Oops, sorry, wrong button, my bad.
What button is that? Vibrate?
Yeah, totally. It's the one that extends and retracts the camera. We're pulled around a little. Hang on, let me give it some more beans.
It's kind of different too.
Got a couple squat lobsters,
Yeah.
brittle stars,
All right, thank
barnacle.
Roger. Still
So
wait.
So I think looking off in the distance in WC upper,
Bridge
I
nav.
think I want to go right.
0 meters.
I want to shoot to the right
065.
side.
Or
Uh,
265.
So you can push all the way in there. Is it? Light years away. Sorry, Brian, what were you saying there? I lost
Saying if you look up in WC upper, you see the set of rocks to the right.
it. Right,
So I think I want to cheat to the right side of the ridgeback.
yeah. Did you step over that way, Chris, did you?
Which way?
This way.
You want me to step up that way?
Yeah,
All right,
we're
I can do that.
gonna go to the right just a bit.
Okay.
Okay, you can go wide video. No idea where the camera is.
Bridge, nav. Go ahead and add 15-meter move to the north. Just add it to the existing move. One 5 to the north.
Paramaricid, according to Asako.
Yep.
Thank you.
Lori, I think we collected one a few minutes ago. It looks very similar to that.
It's gonna drop back downstream a bit here, and then we'll jump off. They go really fast.
How many
this way.
samples do we have now for this dive? Kinlan,
Sorry.
how many samples do we have?
So far in this dive,
Mhm.
we have around 11. Around 11.
What's the max?
Sorry?
What's the max amount of samples can we get?
Oh.
Do you know? Or is this a question for ROV?
Oh, that's a science question.
Oh, okay.
I want to say there's like 21 spaces
Okay.
on this sheet. So I'm going to say 21. Or it could be more or less than that.
All right. Well, thank you guys for tuning in to nautiluslive.org. We are currently exploring a seamount, and we're on the summit, located about 35 miles north of Toby Island.
We have a bit of a conundrum there, Chris.
How so?
Jaden's on one side of the ridge and I'm on the other.
I'm coming.
You'll have to come up a bit, Jaden, and we'll hop you over to the other side. You might have to if you do 20 meters due north, that'd be great.
Ooh, that's a big colony.
All right, we already have quite a few meters in north. It just
Yeah.
hasn't started.
That's all right. I want Atalanta about right here.
Okay. Bridge nav. Add another 20 meters to the north.
We'll put Atalanta off the ridge so we can drop down the wall when we want to.
Gotcha.
Flying with my head out the window.
This is definitely the crab dive too, for the number of crabs we've seen compared to the others.
If I can get it down in here. Still cameras here.
This looks like possibly a J-clade bamboo. And a pretty big one. Try and get some beauty shots of it.
Yeah, definitely. Crab's gonna make a nice guest appearance here.
Can we get lasers off?
Thanks for being so diligent on turning them back on, Jaden. I often forget to ask for it. And it's
Yeah,
nice
me
to
too.
turn them back on immediately. It's super
Always,
helpful.
always good to have them on, unless we're doing a beauty shot.
See if I can get this other camera to work. Am I holding still enough, or am I moving too fast?
That's a you question, Harold.
Sorry, one more time.
Is that ROV stable enough there to get a shot? Or?
Yeah, I'm getting some shots. Got a good one with the crab in it.
I don't know if she's got this one working or not. I'm not seeing a... Might have locked me out.
All right,
Okay.
I think you're good.
Moving on. Interesting, a big one on this side.
So our dive plan says this was a 13 and a half hour dive. Is this true still, or because I know we lost some time at the beginning?
I
can't remember.
We extended a little bit from what we wanted, but we got in the water later than we wanted, so it's somewhere in between. I think we lost. It's going to be, I guess, no, it's still going to be, let's see, we put it in at 8:30, we're going to get out at 11 local, so it's still, yeah, 13, 14 hours.
Okay.
We're recovering this morning, are we?
11 o'clock, I think.
Yeah, plan is 11. Can we zoom this sponge, please?
Right here.
Lori, Pat, if you're watching, this is what we collected a little bit earlier. So take a quick look and let me know in the chat if you think this is the thing you've been looking for.
That's good, thanks. Good there, Brad?
Yeah.
Okay, you can go in.
That current is really going if we have this much layback at
Yeah, it's ripping.
650 meters,
It is ripping.
700 meters.
Brian or Harold, can you answer those questions about how do we use these recordings for like the scientific purpose? Like how does the annotation work, or the process of it?
Yeah, so one of the main purposes of collecting this video is to use it for biodiversity and ecology assessments of these seamounts and deep sea habitats. So what we're doing here is exploring and going somewhere new and finding new cool stuff, but once we finish up here, we take the video back to the lab, and this is something that my lab does at the University of Hawaii. We have a whole group of people who just watches back the video and uses software to annotate the video. And annotation essentially just entails identifying all of the animals that we see and the substrates that we see. And we use lots of different resources to help us do that, taxonomic experts, software tools, and just lots of experience watching these videos.
That has
And
to
then
be
that
really challenging.
data gets served to various places, including OBIS and NOAA. So anyone can use that data for their scientific research to search records of animal occurrences.
Okay, you can turn and hurry, look at.
So, and that data is largely sort of underutilized, and it contains a lot of valuable information. So there's all this effort put into coming out on these cruises and doing these ROV dives and expeditions and going to explore new places, but it's just as important to analyze the data after the fact and go through the video and see what you actually found, because that's where a lot of the scientific discoveries end up getting made, is once you actually crunch the numbers.
So this is the first madrepora I've seen on this dive, with a really nice big sponge community growing on, I guess, the madrepora, since I see madrepora on either side of it. This is a really, really beautiful colony and a great example of why we call these things ecosystems engineers. And they've made extra habitat here, and you've got to see all kinds of associated organisms, crinoids standing on crinoids. I mean, this might be a sponge growing on a coral with crinoids standing on the sponge. All right, thank you.
If you had to guess, how many animals would you say are in the shot right now?
Individual organisms?
Yeah.
I mean, if you included all the small stuff in there, probably approaching 100.
That you could annotate just in that one frame?
And annotate for video less, but if you started like tearing the sponge apart and finding all the polychaetes and everything living in it, but there's certainly a couple dozen things that you'll be able to see in the annotation from that.
All right, Dan, I'm going to go ahead and throw a move in.
Give me a second before you do that. I want to look at.
Okay.
And the annotation takes, I can imagine, hours, right?
Yeah, so our general conversion that we
At the sponge
think
here,
about is
right?
4 hours
star,
of
right?
video, or 4 hours of annotation to 1 hour of video if the video
Wow,
is
that's a long
relatively
Oh, that's
low
a
density.
That's a...
But when we get into higher density areas like this, and especially
that's
areas where we don't have as
okay,
much
push in there.
experience annotating, then it can take quite a bit longer.
Let me square it up here.
Thank you, Harold.
Thank you. Wow.
Should be able to get a good tight shot of that.
New
Wow.
coral does not stand a chance. Looks like most of it's already been eaten.
Yeah.
So this is core livery. This is a sea star eating this primnoid. You can see elements of the sea star's stomach extruded out onto the coral, digesting the coral externally, and then slurping up its nutrients after it's digested back into the body of the sea star. Also, you see a bunch of brittle stars here as well. All right, thank you very much.
All right, Joe.
If we could take a look at this crab, I don't know why it looks purple on this camera. Is it actually purple?
Oh,
Yeah.
Oh, wow.
Yeah.
very
It's
purple.
actually purple.
Very different.
Yeah.
Good eye, Harold.
Yeah, let's get a good look at this.
Yes, please.
All right.
I've never seen a purple crab before.
I'm having
Yeah,
flashbacks here.
This could be potentially another, oh, there's another,
Quasi.
well, there's another, what looks more similar to the Chaseon microniscus, but
It looks
that
partially
one's like
purple.
kind of purple.
Purple
So maybe
haze.
the purple is still Chaseon microniscus? I don't know. Asako says, it's a terrible beast.
So that something is growing on
Or
them that
perhaps
makes them
referring to
purple?
the sea star.
Yeah, we got nothing right now.
Like the other one's half purple.
Yeah. Huh.
Yeah, as tight a zoom as you can give us, because I don't know if it's something growing on there or if it's.
I can
Um,
go for the full,
Yeah,
full zoom.
we'll
light.
send these to Mary Wixton in Texas. He's been our
Whoa,
go-to
that looks so
crustacean
cool.
person.
doesn't look like it's growing, and doesn't look like it's
hold
growing.
on. Let me reposition.
Yeah, it almost, yeah, it just looks like one of those normal Chaseon microniscus Someone just spray painted it purple.
I wonder if it's potentially a color change associated with a molt.
That's
it's getting
possible.
ready to molt.
Come on, ROV, I'll take you
Oh
on.
yeah, is it going to do the thing? These are
Yeah.
really funny. Sometimes when they sense the ROV, they posture like they're going to fight the ROV.
Yeah, we did not see any purple ones on the beta cameras. So this is pretty surprising. And we saw a lot of them on the beta cameras, a lot. And not a single one was purple.
Never seen a crab stumble before.
Well, if it is getting ready to molt, it's potential that it's got reduced motion and sensory capabilities as it gets ready to shed its carapace.
Can zoom out for us.
All right, thank you. We're going to have to consult the
hold that
experts
for
on
a
that
second.
one.
Get ahead of it. Get one more shot.
In certain parts of the North Atlantic, there is a limited commercial fishery for this genus of crabs in the Atlantic version. So this is a potentially commercially exploitable species.
Looks less purple from this angle.
It's still pretty purple.
It's still pretty purple.
I mean, and the Atlantic version of this genus, this crab, is bright red generally, or has that kind of pale yellower color we've been seeing today. But I've never seen the purple version.
Okay. Moving on.
Almost bacon time. Can we quick zoom there, please?
Sure. Go ahead, you can push in a bit for him.
Another absolute beast of a sea star.
Yep. Another coral getting munched. All right, thank you.
Okay. Go ahead.
And then, I don't see it now. Never mind.
Do you know how long it take for the sea star to like devour that whole?
Nope. No idea. There have been several long over lunch conversations about that, and we have no evidence.
Okay.
But it is an interesting question with a lot of very important ecology.
What?
This is that
That's
same...
crazy. Look at that stalk.
Wow, what all is living on
It's
that?
covered in stuff.
That's that same black coral we collected yesterday, but goodness gracious has it got associates. Gooseneck barnacles.
Mushroom
Come on,
corals.
what's
Yeah,
happening?
let's take a good
Come
look
down.
at the stalk.
I don't think they're mushrooms. I think they're
Okay.
anemones.
There's some kind of midwater fish swimming towards us too.
So this is a bamboo coral?
No, this
Or-
is a black coral.
black
Got a
coral.
little
This is
greedy on that north move.
That is
there.
incredible.
A black coral
Yeah.
that that we're struggling to identify. We collected a sample of it yesterday,
Oh, okay.
or 2 days ago. The days are running
Rage,
together.
rage nav, 20225.
But its stalk has become quite the haven for all kinds of things. primarily gooseneck barnacles, but there's squat lobsters, there's hydroids, there's anemones, there's brittle stars.
It looks like a mooring line you would see in a
Yeah.
harbor or something.
Totally does.
Come
There's
down a couple more.
crinoids.
That's amazing.
All right, I think I'd love to get a little bit tighter look at one of the anemones up here, but that's
Roger.
kind of optional if, depending on how much current you got.
We got our nose into it now, so we're good. Just moving the ship a little closer.
So Asako's adding that Scott France once did revisit a colony that had been seen being eaten, and they went back to the same colony 2 years later, and the same star was on the same colony and did not consume the entire colony yet.
Wow. Okay, can push in there a bit.
Yeah, so for the audience, the two lasers you're seeing
Wow.
is a measuring device of 10 centimeters.
Oh, that's really cool. All right, that's probably good enough. Thank you.
That's amazing.
And the white zoanthid. And the coral's doing fine.
I'm on the south
Wow.
side of this thing now.
Well,
you
it's,
know.
I mean, it's alive. It's bending over a bit, but
That's because it's so heavy, it seems like.
it's clearly,
Well,
it's not going down without a fight.
Did you get a picture of this thing, Harold?
Yep, got some stills in the stock. If you want to maybe come out for like a side view.
Yeah, I can do. I can now go wide video.
I'll see what I can do.
Oh, come on, Herc. Biting that tether here a bit.
All right, I think we got it.
Roger. I lost the plot. What side of the hill am I at?
North side.
Oh, where what? There. We are where we want to be.
Yeah.
You can move up west now, I think. So.
Lefterly?
Yeah, but. Something's not jiving here because Atlantis.
Pretty jellyfish.
Atlantis deeper than me? I mean, Atlantis got more under it than me.
Two jellyfish together, very pretty.
Whoa.
Great shot.
Yeah.
You want to drop the lasers?
Yep.
So I'm looking north now. Where's Atalanta?
Thank you for tuning in to nautiluslive.org. This is the 48 shift. We're about to move on
You
to
want
the
to just
next
hang out
group.
here until the
So
watch change at this point?
the watch change will be happening soon. Thank you and have a nice day.
Sure. Somebody's on the cinema camera.
Okay.
Yeah, I'm sure you want to do it. Okay. All right. Let's see what he's got going on for. Sure, I'm just retracting the camera. And looks like 2. Is that right, Noel? Looks like they've only taken 1 still. The Niskin?
Yes, correct.
Okay.
Niskin number 2.
Cool, thanks. Yep.
I can't really see in the Niskins. Well, I guess
Yep, there it
it
goes.
Okay.
Yep.
It's a little dark over there.
Yeah, you're just seeing the flashing light in our junction box.
Okay.
Okay. Yep.
Ready to move?
Well, let's figure out where we're
fish
moving.
right there.
to
Nope,
first.
right there. See that
Yeah.
fish? That might be a viper fish.
Okay. Ooh.
Oh.
Wow, I didn't even go anywhere.
Nope. Bummer. Okay.
We're
So
just
looks like.
moving across the top of this ridge here to waypoint 5.
Oh, I see.
We're just
Okay.
going
Yeah.
west for a while.
We're going west? Okay.
Yep.
You want to poke around up here before we get going, or do they
Let's
finish
just
doing
move
that?
slowly.
Move slow? Okay.
Yeah.
Let's start.
They've had a bit of time up here. They came down apparently, or looked on the other side of the ridge a little bit as well.
Okay.
From the map, it doesn't look like a lot, but a little bit. So.
Yeah, let's start just with a 10 meter and then see how that goes. Things are going to move a lot quicker now because we're only at, like, what are we at, 600 meters?
Right,
700 meters.
700 meters.
So we'll swing a lot faster now.
Okay.
Cool. 10 meters. Good morning, bridge. Can we go 10 meters, bearing 256? All right, thank you.
Here's a couple of these Chaseon crabs right over here on bottom left. This is actually a Hawaiian endemic species, Chaseon microniscus. They're tons. Okay. Well, this will just satisfy my curiosity then, since I was asleep.
Yeah, let's zoom in, please.
Is it shedding, or are there
Molting?
2 of them? Molting, I
There
mean?
are 2 of them.
Oh, no,
Are-
not most.
Are they on a date?
It looks like it. Alright, let's not disturb them any
Intrusion.
longer.
Enjoy.
Okay. Okay, I'll count my
Have
points.
fun. Make responsible decisions. Oh,
Looks like they are.
it's a very romantic time. Oh, good morning, everyone. Just before we proceed, could we do a quick round of introductions?
Sure.
Awesome. So, Ali, Hafaday, and hello, everyone. My name is Amanda Dedicatoria. I'm from the island of Guam. I am a freelance writer and filmmaker, and I'm sitting here with you as your science communication fellow. Could we do the front this time? Could we start with Max?
Yeah, my name is Max Luthi. I am sitting in the video seat, and I'm a video intern on Nautilus.
Oh, that's me. My name's Rai, and I'm sitting in the Atalanta seat today.
Everybody, Josh Chernov, RV Operations Manager for OET, and sitting in the Herc seat.
Good morning, I'm Dan Torriello. I'm a research project manager at the Center for Coastal and Ocean Mapping at the University of New Hampshire, and today I am your navigator.
Awesome, thank you. And Noel?
Good morning, everyone. Ali, my name is Noelle Halter. I am the science coordinator for the Ocean Exploration Trust, and I'm sitting in the data logger seat today.
Thank you.
Good morning, everyone. Onir tutao, which is the Palawan way to say it. My name is Greta Sartori. I am a researcher at the Palau International Coral Reef Center, and for this watch, I'm going to be in science seat number 2.
Thank you.
Ali, my name is Jeff Drazen, and I am a professor at the University of Hawaii. I'm a deep sea ecologist, and I specialize in deep sea food webs and deep sea fishes. And today I am the science watch team lead.
Awesome.
Ali, good morning. Wiltutao, thank you, Greta. My name is Menzang Merid. I work for the Office of the President here in the Republic of Palau. On board, I am the cultural liaison for us. Thank you very much.
Josh, can we zoom on these paranomated sponges? Or
Sure.
Yeah.
I'm just looking if we're going to go... Do we want to kind of stay like right on top, or do you want to kind of be on this like edge?
Yeah, you know, I'm thinking that it might be kind of cool to stay a bit on this edge, but then head off a little bit to the left and kind of cross back over. I'd like to see whether one side or the other is different.
Okay. So I think we'll want to try and put... Where's Atalanta on there?
It's a little cyclotron.
Did you just get hurt?
No, I think this
Oh,
is
there
Atalanta
it is.
here, yeah, it's
Okay.
a small icon.
So we might want to get it a little more south, you know what I mean? So I can be a little more in the middle. Otherwise I'm going to be right along that, like forced along that contour.
Yep. Okay.
So maybe just a little step,
Yeah, we'll
little
move
step south, and then we'll
maybe
keep going west after.
Yeah, 10 meters south.
Sure.
Okay.
Okay, we'll zoom in, please.
It's amazing how they anchor with all those silica threads on the bottom.
Yo, come wide, please.
Well, this is definitely our coral forest. It's amazing. So many primnoids.
Do you have an idea what the small yellow corals are that are scattered?
I
really
do
low?
not. I'm thinking they're... I don't know. We'll have to zoom in on a couple more of them.
Okay, on that one right there?
Sure.
Okay, we'll zoom in, please.
That looks like maybe Enelopsamia. The very, very small yellow fragments. Can we try to zoom in on a couple of these? I'm guessing the previous watch did as well, but we'll
let me
get
kind
a
of
little bit
settle
more.
down here a little bit.
They were populating all that rubble beds on the top of the last seamount, if I'm not wrong.
Mm.
There were,
Yeah.
I think you're right, we collected
some of this
several of
debris,
these cup
didn't we?
cup
Yeah.
corals.
So these were,
no.
these were Enelopsamia, the little tiny yellow ones? Yeah, okay.
Seems to be
Sako Matsumoto is on the chat
I
and
think
confirming.
the bigger yellow one is Enelopsamia.
Okay.
But maybe I'm wrong. I think the small ones, if they're the same that are scattered across the actual benthos, or some kind of little cup coral.
Little cup coral, okay.
Based on the collections that we had last time.
Thank you, Josh.
Okay, we'll come wide, please.
Lots of brachiopod shells.
I was just about to say.
Max's favorite.
Do we want to keep moving south to get over to the other side of this ridge, or move along the ridge ways first? Looks like it gets a bit sharper as we move along the ridge.
Yeah, why don't we just keep going down the ridge a little ways, and then we'll hop back and forth as we get closer to where the contours come together.
Okay. Zoomed in now. If you want to see that.
Yeah. Yeah, so maybe like a west move.
It's another fish in the water column there. We have a lot of midwater animals.
Yeah, just keep coming
Yeah,
along.
let's do a west, 270.
Okay.
Yep.
Some big heterocarpus shrimp. Noel, I understand that our biobox situation is pretty full. Is that
That is
true?
correct.
Okay.
What are you interested in?
Just wondering what my options are.
Gotta know your options.
I don't think any more slurps were attempted.
I understand there may be a jam in the slurp tube.
I think that may be the case.
Oh, really? Maybe that was our trouble
Oh,
the
yeah.
last time we were
I think it's possible that we created
Is
that
that
jam.
is that us? Okay.
Really?
I received no updates on if that was tried.
So it's still just chambers 1 and
there's-
2 that are.
Really? Because like we were successful in the first one, and
Yeah.
and then the second one we just did that tiny little. I just can't see how that would clog the.
Well, I think we should try catching a fish.
Yeah, I don't know if they tried again, but there's nothing reported here.
Huh.
Okay. So samples in slurp chambers 1 and 2 only then. Yes.
Correct.
Okay. Big heterocarpus shrimp. And we've got a comment from the chat that the small yellow corals on the bottom are Aguchasomnia. Aguchasomnia. Little cyclothony fish there. Josh, right on this coral, there's an interesting crab.
Okay.
Climbing up the back there.
Let me see.
Right there.
Jeff, what did you say that genus of crabs was?
Chaceon? Chaceon.
Thank you.
Can we zoom in, please? Is it the crab or is that a...
Yeah, right on the top right there.
Oh, I see it. Okay. Sorry, I was looking at the... Crinoid. Of course it's on this side. Kind of looking downhill a little bit. I could come around the other side, maybe.
Sure.
It would look a little better. Okay, we'll come wide for a sec, please. Just make me spin around to face you.
Oh, there we go.
Okay, let's zoom in, please. Zoom a little more.
Fish.
In the water column?
Yeah.
Yep.
I think it was a squid, actually.
Oh.
That is a lot of brachiopod shells.
Isn't it?
So this is a crab and not a squat lobster?
Squat lobster.
Oh, all right,
Yeah,
okay.
it is.
It looked like...
Yeah, it's probably chirostylid. Maybe munidopsid.
Okay, we'll come wide. Are we good?
Yep.
Okay, we'll come wide, please.
Yeah. Since this guy's going to line up perfectly, let's zoom on the Oreo for a minute.
Sure. We'll zoom in, please.
Did they collect a rock from the top yet?
Such funky dorsal and anal fin spines.
They have such big eyes.
They do.
We collected one that I presume is closer to the bottom.
That's great. Thank you.
Okay, we'll come wide, please.
We got one with May around 1 a.m.
I think that's the one I'm talking about.
Yeah, I think we should get a rock, if possible, from this ridge.
Okay.
At some
Where
point.
would we put that? What do we have for options?
Maybe we can put it with the other rock. In Lambda, I think you were? No, I'm mistaken.
We could put it with the other rock, assuming that they look fairly different.
The other rock is in a small starboard box.
Yeah, the other rock would be very distinctive. It looks like it's like a mushroom-shaped one.
That's right.
So I don't think it can be mistaken with
I'm not sure if we could fit 2 rocks in the small bio box.
Okay.
I don't have a sense of how big their sample was. The rest are corals or sponges.
It was probably.
There's just a small coral.
Oh, can we
There's
look up
something
into the water
in
column
the water
really quickly?
column.
Up and to the right.
can see
more
it
to the,
upper.
there it is, right there. Oh, wow.
Whoa.
Okay, this is a giant heteropod. This is actually a gastropod. Its shell is hanging down on the bottom.
This is incredible.
And the, yeah, oh, this is one's huge too. This is like 30 centimeters long.
Whoa.
These are predators. They're mid-water, so this is an animal that normally would not be associated with the seafloor. And they consume usually other pelagic snails. Their little shell on the bottom is basically vestigial, meaning it's barely used anymore. The animal has eyes. You can see them. They're just a touch bluish on the left-hand side of the animal. And it has that little snout coming out the front of it, and it looks rather innocuous, except it has a producible proboscis, and this thing is like an alien. And it actually sucks other snails practically right out of their shells.
Oh, wow.
Yeah. Little fin for swimming on the other side there is the modified foot.
Can we turn the lasers off?
Yeah. This is great.
Yeah, this is mind-blowing.
Beautiful.
I never heard of anything like this.
I've seen a few of these just snorkeling in blue water, but very small ones, a couple of centimeters in length. I've never seen one of these giant heteropods like this.
And this is a very primitive gastropod shell.
Yeah, absolutely. It's just a cap like a limpets.
Yeah.
Wow.
Wow.
That's so cool.
With
I'm
those
really glad
eyes.
you saw that, Greta.
Oh yeah, I'm really glad I saw this.
Can they move fast?
Thank you, Steel Cam.
I'm not sure. I think they're primarily preying on other pelagic snails like pteropods and things like that don't have huge mobility. So I'm not guessing it moves very fast.
That's
It's like an abstract dolphin.
It looks like an upside down dolphin.
Exactly the same thing.
Yeah,
It looks
sure.
upside down. Actually, when scientists first illustrated these, they had only ever captured them from trawls, and they always put the shell on top because they just assumed it's a gastropod. The shell's on top. It's like a snail. And that's, in fact, not how we see them. They're always like this.
Can we get any on this camera? Do you think
Yeah.
it won't work? Yeah, definitely get a lot of those, if possible.
I don't
See if
understand
I
the
can get a
concept
little
of
closer in there.
this
I'll
shell.
try and get a
What
little
does it-
closer in the DSC.
It's vestigial. It has no purpose. It's
Okay.
leftover. Over evolutionary time, you could imagine these animals would eventually lose them.
Josh, do you think we can take some nice stills of this?
I'm trying to get closer right now.
Thank
yeah.
you, thank you.
Yeah.
So it's version of a gallbladder.
Yeah, there you go. That's a great analogy. Yeah.
Just let
Maybe
me know
not a
I'm not
gallbladder, but
watching
an
the tether
appendix.
at all.
Appendix.
Oh, appendix.
Yeah.
I mean,
Yeah.
I'm
appendix.
good.
Yeah.
so
I knew
cool.
what you meant.
My malacology professor would be going crazy right now.
Yeah, the vast majority of these are less than 10 centimeters. This is massive.
Oh yeah, we're getting some nice stills, I think.
So pretty. It's so graceful the way that
Wait,
it
so
moves.
what is this?
Wait, who's that?
The entire downstairs lounge is freaking
Oh,
out.
hello,
Jen, can
entire
you give your
downstairs
spiel
lounge. This is a
Oh,
thing
sweet. That's a good shot.
that's so beautiful.
It's called a heteropod.
We have no words for it. I mean, heteropod, yeah.
Okay.
So cute, so
Are
cool.
we good? I got a little bit more tether. I'm going to keep, I'll
I
try
can
and get
come
it back.
down.
Can we show it again?
Yeah, I'm trying to get,
So cool.
see
Here
if
it
I can
comes.
get it down there
You're
again.
not below me anymore, so I can come down.
Okay.
What an excellent start to the morning.
Let's see if I can, I'm going to turn just a little bit and see if I can get a little, like, dark background shot.
Yeah.
Looks phenomenal in the lower WCA.
Yeah.
Yeah,
I
it
know.
really
I
does.
hope someone's trying to take stills to that lower camera now, but
There you go.
Does it have eyes? Like, how does it?
Who's controlling the WCA right now?
Rachel.
Would be not in our hands at the moment.
Okay.
But, yeah.
I think we have some cool shots.
Look at it. It's so cool.
Blackwater diving here.
Yeah, I'm just starting to, well, I think we got lots of images. We're just starting to run out of tether now.
Here, I'm coming down a little bit more.
Okay. Yeah, you should be fine. We're just at the very top of this thing, so you can come down pretty low. Okay. Do you want any more zooms of the HD, or?
I think Megan is trying to get a couple more captures of the WCA right now.
Okay.
Yeah.
This is awesome.
Let's wait for that tail to come around and the whole animal to be in profile on the WCA there.
That's cool. That's an all black background.
Wow.
Well, let's definitely keep our eyes open for other midwater animals that are just drifting across the top of this ridge. This is great.
Yeah, I don't have enough tether to go around the other side of it.
I'm gonna send this to my malacology professor back in Italy.
Good.
Okay.
Cool.
Yeah.
They're gonna be so jealous.
With time to get back in contact, let him know what I'm doing.
We saw this in real time. That's amazing. Thank you for spotting it.
All good.
Oh yeah,
That was really cool.
Thanks myself.
I'm gonna come
Jeff Bryant said they saw
starboard.
quite a lot of midwater fish
You got,
last watch, so
almost
cool.
2 ROVs here.
Outstanding. Okay. Well.
Get Jeff all excited.
Oh, there's a giant siphonophore in the water
Oh,
column,
my
and if
goodness.
we're on the midwater theme, we might want to zoom on that too.
Okay,
was on
I'm
my
right
watch
at the tail
list.
end of my tether here, so
Oh
see
my
what I can do.
Bummer.
All right, we can zoom in on that too.
Hello.
Okay, so you just look at the benthic, I look at the steel cam in the water column for
It's
this
going to be
one.
very hard for me not to look in the water column.
Oh my gosh. When I visited,
So, go ahead.
Go for it.
Nope, you go.
When I visited the last ship, there was a lot of. We should get Jeff in here because there's so many fish. But yes, about this siphonophore.
They should have woken me up. Okay, the bells are on the bottom left. You can zoom in on the. If you can zoom, the bells are probably so far
Probably
away.
getting pulled on.
This
Yeah.
thing is many meters long, but seeing the bells is how we would identify what species of siphonophore or what group of siphonophores it was in.
It will come
You can
wide, please.
see all of the thin tentacles streaming off of the main axis of these, and they feed on a lot of midwater plankton, copepods, krill, things like that. Some of these siphonophores get
You want to get
massive,
the wraps out?
tens of meters
well,
in
when I
length. That
pass
one
you,
was
then you
several
can-
meters
Yeah.
in length. Oh, there's a little midwater fish. That's cyclothony.
Cool.
We can skip that one.
Okay, let me get some tether back so I can
Yeah,
actually,
yeah.
like look at something.
Yep.
Let me see it.
Are siphonophores colonial organisms?
Yeah, they're a colonial cnidarian, or like a colonial jelly. And the swimming bells are individual zooids or members of the community at the one
Need
end,
one of them.
and they pump water and help propel the whole animal. And then along its length, you have a combination of colony members that have long tentacles for capturing prey, and others that are actually adapted for the prey to come to. And they're called gastrozoids. They're the stomachs for the whole colony, and they dissolve the prey.
That's so cool. Thank you.
Yeah, some of them are so long, and the side tentacles can spread out in a fan. And some of my colleagues at Mbari call them curtains of death because they.
Oh, no.
Right there. There's small fish on the left. It's not cyclothony. Oh, maybe it is. Oh, it's gone. No worries.
I guess we have to glance at the midwater now. That was so cool. Is that an Oreo?
Yep. There are a lot of Oreos on the seamounts in Palau. We have seen so many of those.
Just you can just turn the. Where are you going to turn to starboard when I come by? Then we could fix it on the recovery. No, we'll wait till I get more ahead.
What gives the corals their color at this depth?
What is the color?
Mm.
Oh, the color of light from.
Like maybe proteins, like we have the zooxanthellae and the shallow water corals that give them the color.
Oh, I see. What's giving the pigments to
Yeah,
the corals?
yeah.
Yeah, I don't actually know. Excuse me. I have no idea why a coral at depth would be yellow, for instance.
Traffic safety.
Yeah.
Slows down the current.
So, Ken Sudak and I were contemplating that some of these colors may have functions that we really can't imagine because we're not seeing the low light environment that these organisms actually are living in. And there may be interactions between bioluminescence and some of their colors.
Mm-hmm.
So Victorigorgia is bright purple. Why? There's not a lot of bright purple things down here. The Anopsamia is yellow. And those are sort of figuring out whether there's a ecological role to those colors is really going to require doing some work with the sort of starlight cameras and the ability to see bioluminescence.
I see.
Yeah, let's do another 10 meters just like along that ridge, like whatever that bearing is. Like so Herc's going along the, or yeah, following that top, contour, yeah.
Can we check out that fish right there?
Sure. Okay, we'll zoom in, please. I think we need lasers back on if you want them to.
It's a rattail fish. Oddly short body.
Looks to
we can
not
push in
be
some more.
doing
it. Josh, we're gonna try to collect this fish.
Oh yeah, okay.
Yeah, it's this is a fish not doing very well.
Okay, we'll come wide, please.
And
can you set up the jar?
Yeah.
And grab the.
Looks like a little too shiny to be the Kumbo we've seen in the past.
Let's stop the ship here.
Oh, we want to attempt to slurp this.
Cut the
yeah, we should try to index into place 3 and maybe just run the pump for a minute to see what happens. Oh, yep, there went our coral.
Okay, that's it.
So go to 4. Go to 4.
Our whip coral is now in 3.
Yeah. So let's go to 4.
Okay, yeah. Sample jar 4.
Yeah. And then I'm gonna tilt the.
That was good, huh? Lucky.
I'm still, Jeff, you still got eyes on it. It kind of
I do.
just above
right
the lasers,
there.
right?
Yep.
Yeah. Okay.
slurp.
Never tracked. Well, of course it goes like under
I know.
overhang.
And there's a cut-through eel on the bottom, and when this thing gets down to the bottom, that cut-through eel is going to eat this fish. There's something wrong with this fish, so.
It's like in a little alcove.
Yeah.
Okay, I've got the slurp.
Okay, I'll porch back a little bit.
Yeah, great.
Let's get another zoom on the fish. It looks like it's kind of drifting outwards now.
Okay,
So.
we'll just hold the arm there for a sec.
Yeah.
Extend out. Okay, Max, let's zoom in, please.
So Jeff, would you have a way to tell what's happening to this fish?
It's possible with a necropsy.
Is that?
Yep, that's it.
Is it?
Yep, it is.
It's just kind of like turned towards you.
Yeah, I just want to get a good profile shot. And one of the main things we need to do is be able to get good video, and we did get some good video before, so that we can try to identify these fishes from video. So we need the physical specimen with the video. That's the most valuable thing.
That's not looking too happy.
No, we're going to be able to collect this fish. It's missing its tail tip. It's super shiny. It looks like almost like Ventra fossa, but the nose isn't right. Oh, that's good. Really long dorsal fin.
Is that like
really
a parasite
nose.
or something on like just above its eye, or?
I don't think so.
No, don't think so. Okay, just looks kind of odd.
Okay, we can collect this fish now.
Okay.
Thank you.
Come wide, please. And let me bring up my...
And that's a conger eel. Okay, not... Yeah.
Okay, let me get in a better spot.
Yeah.
I gotta slow here, real slow.
Yeah, there's nothing majorly outwardly looking wrong with this fish, but obviously it is on its way out.
Okay, I turn on the suction.
Hey Josh, are you okay with me zooming in and out on the cinema cam?
Yeah, that's totally fine.
Okay, thank you.
Okay, should hopefully flow in water. Can we just do like a partial zoom and keep the porch in view?
It's pretty much on the ground now.
Thank you.
Let me know if you want me to just reach out.
Yeah, I think you can try and go for it. Just touching the bottom there.
Yeah. Suction's on?
Yep, there it goes.
There it goes.
Yep.
Well, that was
Okay, come wide, please.
Wait, watch if it gets in there.
Yeah,
There it is.
Got it.
Oh, that's fantastic.
Okay, and then
Thank
I'll
you
rotate
both.
the jar.
Rat tail number 2 for the expedition.
Oh, that's pretty big.
Yeah, so the suction is working just fine. Okay.
I think you just needed the
little
jar and the
yeah. Okay. We're full wide. Max, are we full wide?
We are full wide.
Okay, thank you. Okay, I'm gonna porch in. I'm gonna turn this down. Okay. Okay, let's, why don't we start that 10 step again?
Yeah, The urchin, Histocidaris.
It's an urchin.
Yeah, we've seen a couple of those.
Yeah, like yesterday night too.
Yep.
Let's do another 10.
This is a slightly different primnoid on the left. If we can zoom on this one here. Most of the fans are just one layer, and this looks like to be multiple layers.
We'll zoom in, please.
That's beautiful.
It really is.
Settle down here. We can go for a polyp shot here. Okay, we can go in more, please.
So these primnodes have lots of spikes, right? All around the polyps and up and down the stems. Is that just to prevent predation? Is that the main thought?
I presume so, yeah. Is to help. I mean, you know, of course, their predators are things like sea stars and that sort of thing. So I think that helps. That's great.
Okay, come on,
Thanks.
please.